So it's understanding that dynamic, changing environment that is the key challenge right now. And Mike might want to tell you about some, he's been one of the most measured people in the world because we've begun to actually measure his proteome, his metabolome. I don't know if you want to mention that.
So you test on yourself.
Yeah, we do. Okay. So, I mean, really where the field is going, we're trying to study our genes, our DNA, to be able to predict human disease. So this is how the project started. We wanted to actually start studying DNA of healthy people and try and predict disease risk. So we actually started on me as the guinea pig because that seemed pretty safe and something I could do without worrying about traumatizing anyone besides myself. So we basically started by actually determining my DNA sequence and then trying to predict the diseases I was at risk for. At the same time, we decided to do a very detailed profile where we would actually look at all the molecules in my blood, my proteins, my metabolites, actually my RNA, which encodes for the protein. So we're actually studying all of my molecules. We make billions of measurements right now on my blood and actually in other parts of my body to be able to see in detail. It's like getting an IMAX movie of your molecular physiology. And we're doing that so that we can see what happens when people's disease might come on. Now, what happened in my case was we sequenced my DNA, and surprisingly, I was predicted to be at risk for diabetes. And as we were going along in the study, about 9 months in, it turns out I got diabetes. My sugar shot through the roof, and we caught that because we were making these billions of measurements in my blood so we could see my glucose go astray. We actually caught it right away, so I in fact changed my lifestyle, meaning I started eating better and I started exercising more, and I gradually brought my glucose under control. But we've been doing all this so we can both understand in detail the physiological changes when someone gets disease, but also the goal is to be able to catch disease early and ideally predict disease and prevent it before it even happens in the first place. That's where we see genomics going in the future.
So it worked in this case, a good case study. We have one more slide in this series. Can we see that next slide, please?
Yeah, in a sense, that's me. So that you can think of that as me in the middle, measuring my DNA in the bottom left part there, and then we're measuring all my proteins and my antibodies, which are things that are my defense system against viruses and bacteria and evil pathogens. RNA, which encodes for proteins in the upper right there, and metabolites are on the left. So we're actually making measurements of all of these things. Again, many billions of measurements on all my molecules in my blood, and actually we do it in my urine and other places as well.
You know, this sounds like science fiction, but this is what we're headed towards in the clinic. Because as a patient comes in right now, we always draw blood tests, but it's very primitive. It's just a couple of things that we know how to measure because we've known how to measure them for 50 years. I think the importance of what Mike was just talking about is this gives us a window into the kind of measurement we're going to be making of patients in the next 20, 30 years. We're going to have a much higher resolution of the dynamic changes that this patient's physiology is undergoing. And the great thing about this story is we're going to catch disease early when it still might be intervention compatible. And that's, again, as a physician, that's the most exciting thing is I don't want a patient to come in where everything has already happened and I'm just in response mode. I'd like to be able to make measurements in real time so that we can proactively alter the course of a disease and maybe even nip it at its bud. So this is very exciting, and I don't think you should think about it as just something that Mike gets to do. I think we're all going to be able to do this.
I understand that you're also involved in something called personalized medicine, in that some people react differently to different types of medication, and that can be predicted by their genome, so you could tailor medication depending on a person's genetic traits. In fact, I think we have a slide about that. Can we see that slide, please?
Yeah, so if I may, what you have in the middle there is the population, the diverse population of humanity. And when I give a medication, I have a rough idea what might happen to the patient. But let's be honest, I think we've all had an experience where the drug doesn't work exactly as planned. Sometimes it'll work exactly as we hoped. Sometimes there won't be any effect at all. Sometimes there'll actually be a toxic effect instead of any kind of efficacious effect, and sometimes there won't be, there'll be a toxic effect as well as a good effect. And you can see all those different combinations around the side there. I think the important thing is, in your genome are variations in your DNA that allow us, in many cases, to predict whether a certain medication is more likely to be toxic for you or more likely to work well for you. And this is possible because we understand how drugs work, and drugs are metabolized usually by the liver or the kidney. And if you have genes that are in the liver or the kidney that have small variations, you may, for example, absorb a drug faster than somebody else or slower. That difference in absorption will lead to a different experience of the drug. Similarly, the drug, when it reaches its target, that is to say the protein that it interacts with, there may be variations in that target that make the drug bind either more tightly or less tightly, and that could lead to a different experience of the drug. There are drugs that everybody in the world has tried. Codeine is one commonly known drug, where there is a huge variation in the population of whether codeine is effective or not. And most people don't realize that variation is because of their genetic background.
What is the ultimate thing you're trying to learn about in genome research? What is the holy grail of all this?
Well, in my mind, the goal would be to know the genome so well that when a child is born, based on their DNA sequence, we could predict all of the diseases that they're at risk for. It wouldn't be deterministic, it would be probabilistic. And what I mean by that is the following, that based on a genome sequence, you'd have a certain chance of getting a certain kind of disease. And we would like to know that, again, the genome so well that if there were a change that came out, you would be able to make that prediction. And that would be my goal.
Is there a possibility of fixing somebody's genome so they're no longer vulnerable to that disease?
There is. There's an entire field called gene therapy or gene intervention, where we try to introduce genes that have been mutated to the point where they're not functioning and they cause a disease. Sometimes you can reintroduce using a variety of viral or man-made, human-made vectors, introduce a fixed-up version of the DNA into enough cells that the organ starts functioning appropriately. And we're looking at this for some very rare diseases where there's a known genetic variation. And there are trials now in humans that are quite promising. So I think that in our lifetime we may see focused correction of a severe human disease.